Infectious nature of nucleic acid was discovered by. Infectious nature of, this is all history of viruses. Infectious nature of nucleic acids was discovered by Gerer, not Frankel, not Twart, bacteriophages, Delbruck, lytic cycle in viruses. So, Gerer. Zymophages are parasitic on. Zymophages are parasitic on. Based on the host on which they attack, viruses are named with different names. Phytophages means attacking plants. Zoophages means attacking animals. Cyanophages means attacking blue-green algae. Attacking blue-green algae. Gymophages means attacking yeast cells. Bacteriophages means attacking bacteria. Attacking bacteria. Coliphages means attacking Escherichia coli. So, zoophages, cyanophages, mycophages means bacteria attacking fungi. So, on which a virus is attacking, different names are given. Phytophage attacking plants, zoophage, like that. The question given is the virus attacking yeast cells. Virus attacking yeast cells is zymophage, fungi is mycophage, cyanobacteria is cyanophage, virus attacking actinomycetes is not discovered. So, yeast is the answer. The smallest one regarding the structures: ribosome, virus, yeast and Passerella. In this, the smallest one is ribosome. Ribosome is smallest, next is virus, next is Passerella and finally yeast. So, first is the action. This is the smallest virus, ribosome. Smallest virus is cyanophage, bacteriophage, zymophage, mycophage. Just now I told cyanophage is attacking blue-green algae, bacteriophage is attacking bacteria, mycophage is attacking fungi, zymophage is attacking yeast cell. So, the smallest virus known is zymophage. This virus can be seen under a compound microscope. This virus, because vaccinia virus is supposed to be the largest virus. So, vaccinia virus is the largest virus, but equal to the smallest bacterium called Passerella. So, Passerella pestis is regarded as smallest bacterium. The largest bacterium, the largest virus, vaccinia virus, it is the largest in viruses, but it is exactly almost equal to the smallest bacterium called Passerella. So, because variola, zymophages, cyanophages are very small in nature, the virus can be seen under compound microscope is vaccinia virus. Arrangement of capsomeres of virus. Arrangement of capsomeres of virus determines infectious nature of virus, host specificity, symmetry, size. Not infectious nature of viruses, not host specificity, symmetry and size. So, based on capsomeres, arrangement of capsomeres, viruses show two types of symmetry. One is helical symmetry, the other is cubical symmetry. Hemical symmetry means it is found in tobacco mosaic virus. Why? Because here capsomeres are arranged around the nucleic acid RNA as spiral staircase or are arranged as in a spiral manner. Cubical viruses are adenoviruses. Here capsomeres resembles an equilateral triangle. Every capsomere is an equilateral triangle. 20 such equilateral triangles are arranged in a cubical manner. 20 such equilateral triangles are arranged in the form of a cubical manner. This is itself is called icosahedron symmetry. It is very popularly called icosahedron symmetry. So, helical viruses, helical symmetry is shown by tobacco mosaic virus. Here RNA is surrounded by capsomeres which are arranged like a spiral staircase. Cubicle is found in adenovirus. Capsomeres, each capsomere is an equilateral triangle. 20 such equilateral triangle faces are arranged in a cubical manner, which is called icosahedron symmetry. There is another one symmetry called binal symmetry. Binal symmetry is seen in T2, T4 even phases. What is binal symmetry means? Every bacteriophage has two parts, head and tail. Head shows cubical symmetry, tail shows helical symmetry. So, two types of symmetry are seen. Head is showing cubical symmetry, tail is showing helical symmetry. This type of symmetry is popularly called binal symmetry. So, arrangement of capsomeres in virus results in studying the symmetry in viruses. So, whenever we want to study the symmetry in viruses is of two types. One is helical symmetry, the other is cubical symmetry. Helical symmetry is found in tobacco mosaic virus where capsomeres are arranged as a spiral staircase. Cubical symmetry is found in adenoviruses where the capsomeres, each capsomere is an equilateral triangle face which ultimately arranged like a cube. Binal symmetry is found in bacteriophages. Head shows cubical symmetry in capsomeres, tail shows helical symmetry in capsomeres. So, symmetry is arrangement of capsomeres of shape of rhabdovirus. Rhabdovirus, special viruses, the shape of this in plants, in animals is bullet shape. The same shape of this virus in plants is rod shape. So, bullet shaped viruses are rhabdoviruses, but their bullet shape is seen in animals, but the same rhabdovirus is rod shaped in plants. So, shape of rhabdovirus is spherical, rod shaped, oval shaped, bullet shaped. Bullet shaped in the case of animals and rod shaped in the case of plants. So, for this answer is 2 and 4 will come. Next, the shape of the largest virus. Largest virus, just now I told vaccinia virus. The shape of the vaccinia virus is rectangular, brick shaped. It is popularly called brick shaped virus. It is not quadrangular, it is not ellipsoidal, it is not oval, it is rectangular or brick shaped. Arrangement of capsomeres in the form of a staircase is seen in. Arrangement of capsomeres in the form of staircase is helical symmetry. That means the single stranded RNA, the genetic material in tobacco mosaic virus is single stranded RNA. The single stranded RNA is enclosed in a special core whose diameter is around 4 nanometers. This core is surrounded by numerous protein bits called capsomeres. The capsomere number here is 2130. 2130 capsomeres are present, and these 2130 capsomeres are arranged in the form of a spiral staircase. Every capsomere shows 158 amino acids. In every capsomere, the total number of amino acids is 158, and this single stranded RNA contains 6500 nucleotides. 6500 nucleotides. So, the capsomeres are arranged in the form of a spiral staircase in the symmetry of tobacco mosaic virus. The number of capsomeres is 2130. Every capsomere contains 158 amino acids, and every DNA contains, single stranded RNA contains 6,500 nucleotides. So, arrangement of capsomeres is in the form of a staircase is seen in tobacco mosaic virus. Adenovirus, the arrangement of capsomeres is in the form of a cube, where each capsomere is an equilateral triangle face arranged in a cubical manner. So, 14th question answer is 1. Each phase of cubical virus. Each phase means each capsomere of tubercle virus is triangular. It is an equilateral triangle. How many such phases? 20 phases are arranged in a cubical manner. This type of symmetry is called icosahedron symmetry. So, it is not rectangular, it is not triangular, it is not quadrangular, it is not oval, but it is triangular, equilateral triangle phase. Second option. Both helical and cubical symmetry are seen in these viruses. Every bacteriophage has a structure like this. It shows a hexagonal head and a narrow tail. Hexagonal head, narrow tail. In between head and the tail there is a special structure called collar. At the base of the tail there is a special portion called end plate or tail plate. End plate or tail plate gives rise to 6 spikes. Every spike produces a tail fibril. So, in total 6 tail fibrils are present. The core of tail is empty, whereas the core of head is filled with double stranded DNA. Now, this capsid protein coat, capsomeres are arranged in helical manner, whereas here the capsomeres are arranged in cubical manner. So, because the bacteriophage shows a head where capsomeres are arranged in cubical manner and a tail shows capsomeres arranged in helical manner, the symmetry in these is called binal symmetry, because tail shows helical symmetry, head shows cubical symmetry. T2 bacteriophages. Polypeptides, lipids, carbohydrates and polynucleotides all are seen in influenza virus. Influenza virus is an animal virus. Every virus normally shows a genetic material at the center, either DNA or RNA. No virus is with both DNA and RNA. It is surrounded by a special protein coat called capsid protein. The influenza speciality is it has another coat called peplose. This peplose is made up of many bits called peplomeres. So, because of peplose, this is influenza virus. Because of peplose, it is supposed to be containing above proteins some other important aspects. What are the some other important aspects? Polypeptides, lipids, carbohydrates and polynucleotides are seen in not in TMV, not in T2 bacteriophage, not in cyanophage, but in influenza virus. Influenza virus is an animal virus where the capsid is again surrounded by another special layer called peplose, made up of peplomeres. Arrange the following pathogens in a sequence that have only nucleic acid and protein coat, only protein covering nucleic acid, capsid and peplose. Only for this answer is 1, 3, 2, 4. Only nucleic acid is citrus exocartosis virus. Then 3, TMV. This is nucleic acid and protein coat is tobacco mosaic virus. Then only protein coat is scrapie disease pathogen, and nucleic acid, capsid and peplose is influenza virus. Viruses based on genetic material divided into 4 types according to this. One is virion, which has capsid protein coat and nucleic acid. Viroid, it has only nucleic acid without coat. Prion, this has only capsid protein coat without nucleic acid. And peplose is found in influenza virus. For this many examples TMV, For this, citrus exocartosis virus and potato spindle tuber virus. For this, scrapie disease in sheep and mad cow disease in cattle. Scrapie disease in sheep and mad cow disease in cattle and other animals. So, virion, viroid, prion, peplose. Based on this concept, this question is given. So, arrange the pathogens in a sequence that have only nucleic acid is viroid. Nucleic acid and protein coat is tobacco mosaic virus. What is viroid here? Citrus exocartosis virus. Only protein covering is scrapie disease in sheep and mad cow disease in cattle, which is prion. Then covering nucleic acid, capsid and peplose is influenza virus. 1, 3, 2, 4. Arrange the following discoveries in a sequence: crystallization of viruses, nucleic acid and protein coat composition of viruses, rabies vaccination, contagium vivum fluidum. First is rabies vaccination, Louis Pasteur. Then contagium vivum fluidum by Beisering. Then crystallization of viruses by W.M. Stanley. Finally, nucleic acid as protein composition of virus, protein and nucleic acid, Borden and Pyrie. So, first Louis Pasteur rabies vaccination, then Beisering contagium vivum fluidum, then W.M. Stanley crystallization of viruses, TMV in a pure crystalline form, nucleic acid and protein composition of viruses by Borden and Pyrie. So, according to scientists and their ages, first Louis Pasteur, then Beisering, then Stanley, and finally Borden and Pyrie. So, answer is 3, 4, 1, 2. Arrange the following viruses with increase in complexity of their symmetry due to arrangement of their capsomeres. Viruses with increase in the complexity of their symmetry due to the arrangement of their capsomeres. First is 3, potato spindle tuber virus, because here you do not find any capsomere. So, 0. Potato spindle tuber virus is a viroid where capsid protein coat is absent. Then 2. 2 is tobacco mosaic virus, 2130 are present. Then 1, TMV, tobacco mosaic T2 bacteriophage has binal symmetry. Then adenovirus has many capsomeres. So, 3, 2, 1, 4, second option. Now, this is a matching column: cyanophages, phytophages, zoophages, bacteriophages. Cyanophages, blue-green algae. Cyanophage means virus attacking blue-green algae. Phytophages. Phytophage is virus attacking plants. Tobacco mosaic virus is a phytophage which attacks plants. Zoophages. Zoophages means HIV, human immunovirus, the virus which attack animals and cause AIDS, that is zoophages. Bacteriophages. Bacteriophage is T2 bacteriophages. T2 here zymophages are not given, so yeast virus is wrong. So, cyanophages is attacking blue-green algae. Phytophages is tobacco mosaic virus which attacks tobacco plants. Zoophages is HIV, human immunovirus responsible for AIDS, and bacteriophage is T2 phages. T2 phages. So, answer is 3, A3, B4, C2, D1. Poliovirus, adenovirus, rhabdovirus, vaccinia virus. This matching is according to the shapes. Poliovirus is spherical, 3. Adenovirus is polyhedral, 4. Rhabdovirus is bullet shaped, 5. Vaccinia virus is rectangular, 1. So, A3, B4, C5, D1. Poliovirus is always spheroidal. Adenovirus is always polyhedral. Rhabdovirus is always bullet shaped. Vaccinia virus is always rectangular. So, answer is 2. This is 2 correct items. Ivanovsky discovery of viroids tobacco mosaic virus is wrong. Discovery of viroids is not done by Ivanovsky. Ivanovsky has done only irises are filterable agents. Franklin Conrad ultrastructure of viruses tobacco mosaic virus is correct. Ultrastructure of viruses, and that too genetic material in plant viruses is RNA is by Franklin Conrad. Delbruck viral replication by bacteriophage is correct. Lederberg transduction tobacco mosaic virus is wrong. Lederberg is associated with transduction, but not tobacco mosaic virus. It is Salmonella typhimurium. So, Ivanovsky discovery of virus itself is wrong. TMV is correct. He proved that tobacco mosaic virus is filterable. Franklin Conrad ultrastructure of viruses, tobacco mosaic virus is correct. Delbruck viral replication, bacteriophages is correct, but Lederberg transduction is correct, but not tobacco mosaic virus. So, 2 and 3 are correct. Second option. Next, virus host genetic material. T2 bacteriophages, bacterium RNA is wrong. RNA is wrong. DNA must be present. So, D2 bacteriophages attacking bacterium is correct, but genetic material RNA is wrong. Tobacco mosaic virus, Nicotiana tabacum, single stranded RNA is correct. Tobacco mosaic virus, Nicotiana tabacum, single stranded RNA is correct. 5x174 bacteriophages, bacterium, single stranded DNA is correct. Single stranded DNA is found in 5x174 bacteriophages and M13 bacteriophages. Influenza virus, man, DNA is wrong, because plant viruses with DNA are Dahlia mosaic virus and cauliflower mosaic virus. Animal viruses with RNA are influenza virus and polio virus. These two are RNA containing viruses. So, influenza virus attacking man is correct, but genetic material is not DNA, it is RNA. 5x174 bacteriophages attacking bacteria, single stranded DNA is correct. Tobacco mosaic virus, Nicotiana, single stranded RNA is correct. T2 phages, bacterium, RNA is wrong. So, the correct combinations or items are 2 and 3, that is second option. So, I will write some important. Normally viruses show either single stranded RNA or double stranded DNA. Normally plant viruses have RNA, but in cauliflower mosaic virus and Dahlia mosaic virus, the genetic material is DNA. Normally animal viruses have DNA, but influenza virus and polio virus has RNA. Some viruses have double stranded RNA like Rio virus, wound tumor virus, blue tongue virus, rice dwarf virus, maize rough dwarf virus, and 5-6 bacteriophages has double stranded RNA as genetic material. Some viruses have single stranded DNA. Just now I told 5x bacteriophages and M13 bacteriophages. So, here T2 phase, bacterium RNA is wrong, it must be DNA. TMB Nicotiana, single stranded RNA is correct. 5x174 bacteriophage, bacterium, single stranded DNA is correct. Influenza virus, man is correct, but not DNA, it is RNA. So, answer is 2. 2 and 3 are correct. Now, match the following: single stranded DNA, double stranded DNA, single stranded RNA, double stranded RNA. Single stranded DNA is found in M13 bacteriophages 5. Double stranded DNA is found in bacteriophages 4. Single stranded RNA is found in tobacco mosaic virus 3, and double stranded DNA is found in RNA is found in blue tongue virus. So, answer is 5, 4, 3, 1. Prion shows only capsid protein coat, it does not have any DNA. So, no question of comparison with single stranded or double stranded, either RNA or DNA. So, single stranded DNA M13 bacteriophage, double stranded DNA bacteriophage, single stranded RNA tobacco mosaic virus, double stranded RNA blue tongue virus A5, B4, C3, D1. This is a non-living character of viruses. Presence of polynucleotides, growth outside the host, and no response to external stimuli. So, no response to external stimuli. Only 3 is correct. 1 and 2, 2 and 3, 1 and 3 are wrong. Only 3 is correct. Fourth option. Technique used in virology are ultracentrifugation, X-ray crystallography, compound microscopy. In order to discover viruses, in order to study various viruses and other things, the technique widely used is ultracentrifugation and X-ray crystallography. So, 1 and 2 will be the answers. Bacteriophages show helical symmetry, icosahedron symmetry, zygosymmetry. Helical symmetry in the tail, where capsomeres are arranged in the form of spiral staircase. Icosahedron symmetry in the head, where the capsomeres are arranged in a cubical pattern. So, answer is second, 1 and 2. Nucleic acid is infectious in tobacco mosaic virus, potato spindle tuber virus, prions. Potato spindle tuber virus shows only nucleide without protein coat. Just now we discussed exocartosis, citrus exocartosis virus. Potato spindle tuber virus has only DNA or genetic material without capsid protein coat. Prions have only capsid protein coat without genetic material. So, tobacco mosaic virus, the genetic material is infectious, which cause diseases. Potato spindle tuber virus, only genetic material is present, which is also causing diseases or infectious. Prions protein coat is infectious. So, nucleic acid is infectious in tobacco mosaic virus, potato spindle tuber virus, 1 and 2, second option. Infectious particle has only nucleic acid in. Infectious particle has only nucleic acid in citrus exocartosis virus. Rice dwarf virus is double stranded RNA. Tungro virus causes what we called rice tungro disease. So, citrus exocartosis virus. Just now I told virion is complete viral particle with nucleic acid and protein. Any virus. Viroid is only nucleic acid without protein coat. Citrus exocartosis virus, potato spindle tuber virus. Prion, it has only infectious protein coat, nucleic acid is absent. It also causes diseases like scrapie disease in sheep, mad cow in cattle. So, here the answer is prion. So, what is the question asked? Infectious particle has only nucleic acid. Infectious particle has only protein means scrapie disease in sheep and mad cow disease in cattle, but infectious particle has only nucleic acid means citrus exocartosis virus. Rice dwarf and tungro are wrong. So, answer is 1. Capsid of virus helps in protecting the nucleic acid is correct. Transferring nucleic acid from host cell to other is correct. Taking over the functions of host cell is wrong. Capsid of virus helps only in protecting the nucleic acid. Sometimes it helps in transferring the nucleic acid from host cell to other. Taking over the functions of host cell is wrong. So, protecting nucleic acid is correct. Transferring nucleic acid from host cell to other is correct. So, first option 1 and 2. Plant virus with DNA as genetic material. Plant virus with DNA as genetic material is Dahlia mosaic virus, rice dwarf virus, cauliflower mosaic virus. Normally, plant viruses has RNA, animal viruses has DNA. Plant viruses with DNA are Dahlia mosaic virus and cauliflower mosaic virus. Animal viruses with RNA are influenza virus and polio virus. So, plant viruses with DNA as genetic material are Dahlia mosaic virus and cauliflower mosaic virus. 4, 1 and 3 is the answer. Fourth option. Citrus exocartosis virus is not a virus. It has capsid protein coat. So, a complete viral particle, if it is present, then only it is called as a virus. It is simply called as viroid. It is not a virus. So, citrus exocartosis virus is not a virus is correct. Why it is not a virus means because it does not have any capsid protein coat. So, A and R are correct. R is the correct explanation of A, first option. Viruses are neither living nor non-living. either living or non-living. Outside the host they are non-living, inside the host they are living. So, that is correct. They have ergastric substances and cytosol is completely wrong. Protoplasm itself is not there. How come cytoplasm is present and ergastric substances are present? So, A is true, R is false. Viruses are neither living nor non-living is correct, because outside the host they are non-living, inside the host they are living and infectious. They have ergastric substance and cytosol is wrong. So, A is true, R is false. Vaccines for viral disease was first discovered by Jenner. Vaccine for viral disease was first discovered by Jenner is correct. First vaccination that was discovered is smallpox vaccination. Vaccines are used to prevent viral diseases in plants. Vaccines are not used to prevent viral diseases in plants. They are used to prevent viral diseases in animals and human beings. So, A is true, R is false. Viruses are extracellular parasites. Viruses are host specific. Viruses are extracellular parasites means they are parasitic outside the host. Impossible. Outside the host they are non-living and crystalline. So, they are not extracellular parasites, but viruses are host specific. Definitely phytophages, zoophages, cyanophages, bacteriophages, coliphages, mycophages, all these names are given according to viruses host specific. They attack only particular host. So, viruses are extracellular parasites are wrong. They are not extracellular parasites, they are intracellular. Inside the cell only they are living, outside the cell or they are completely non-living. Viruses are host specific is perfectly correct. So, A is false, R is true. Viruses are too small to see under light microscope. Definitely they are very small. Viruses are made up of nucleoproteins. If you see a virus structure, it shows a central genetic material and a capsid protein coat. This genetic material is either DNA or RNA. This capsid protein coat is called capsid protein coat, which is made up of small bits called capsomeres. Based on that structure, viruses almost resemble ribosomes. Ribosomes are also nucleoproteinous structures. So, ribosomes are also nucleoproteinous structures. They help in protein synthesis. So, structurally every virus resembles a ribosome. So, viruses are too small to see under light microscope. They cannot be observed under light microscope. Viruses are larger than ribosomes, but smaller than bacteria. So, viruses are too small to see under light microscope is correct. Viruses are slightly larger than ribosomes and definitely smaller than bacteria is correct, because they are slightly larger than bacteria and slightly larger than ribosomes and smaller than bacteria. They cannot be seen under light microscope. They have to be seen under compound, not even compound, electron microscope. So, A and R are correct. R is the correct explanation of A, first option. TMV has helical and adenovirus has cubical symmetry. TMV, tobacco mosaic virus, has helical symmetry. Adenovirus has cubical symmetry. Arrangement of capsomeres is different in TMV and adenovirus. Just now we explained tobacco mosaic virus, adenovirus. Tobacco monas virus is a rod shaped virus which is opened on both sides. It was first crystallized by Stanley, but the structure was explained by Frankel or Franklin's. Tobacco mosaic virus shows a central core of DNA surrounded by capsid protein coat made up of capsomeres which are arranged in the form of a spiral staircase. Adenovirus shows cubical symmetry. Cubical symmetry shows icosahedron nature. Icosahedron nature. Here every capsomere is an equilateral triangle. Every equilateral triangle arranged in a cubical manner, and every equilateral triangle How many such equilateral triangles? Around 20 equilateral triangles are arranged in a cubical manner. 20 equilateral triangles are arranged in a cubical manner. So the question here, assertion reasoning is TMV has helical and adenoviruses has cubical symmetry is correct. Arrangement of capsomeres is different in TMV and tobacco. Definitely, helical means capsomeres are arranged in a spiral staircase manner. Cubical means capsomeres are arranged in a cubical manner. So A and R are correct. R is the correct explanation of A. Influenza virus has capsid and peplose. Influenza virus has capsid and peplose. Just now I explained. Influenza virus is an animal virus with RNA. So influenza virus is an animal virus with RNA. It has a genetic material RNA at the center surrounded by protein coat. So RNA genetic material surrounded by protein coat. Caps. Another special layer is present around protein coat called peplose. Peplose is made up of special bits called peplomeres. Capsid is made up of special bits called capsomeres. So this RNA is surrounded by capsid protein coat made up of capsomeres and peplose peplomeres. So influenza virus has capsid and peplose. Correct. All RNA viruses has envelope is wrong. All RNA viruses does not have peplose envelope because tobacco mosaic virus is also a RNA virus. It does not have any peplose. So A is true, R is false. So this is about some of the questions regarding viruses, some of the questions regarding bacteria. So bacteria we discussed completely with almost all important aspects and other things. Viruses we discovered only up to symmetry. Symmetry means first structure of virus, then shape of the viruses, then how viruses attacking the animals or other things. Then we discussed about the chemical nature of viruses, which virus contain capsid and nucleic acid, which virus contain only nucleic acid without capsid, and which virus contain only capsid and without nucleic acid, and what are the viruses which contain peplose, and what are the viruses which have double stranded DNA, single stranded DNA, double stranded RNA, single stranded RNA, and also we discussed symmetry. What is symmetry? Based on the arrangement of capsomere symmetry. Cubical symmetry, helical symmetry, binary symmetry. In the next class we discuss remaining. That means structure of tobacco mosaic virus, bacteriophage, then replication of viruses, lytic cycle, lysogenic cycle. Then we discuss about transmission of viruses, control of viral diseases, and if possible we will start plant physiology. So next class we discuss the remaining viruses and plant physiology mostly up to water relations, that is up to transpiration.